So I'm waiting for it to confirm. There, it's confirmed on my side. Okay, so remember the rules, guys. Your mic needs to be muted at all times unless you have a question. So for example, now I have the slides on my screen, so I can't see anything that is happening on your side. So in order for you to ask a question or to get my attention, please just unmute any second and then ask me the question, because now I can't see who's raising a hand, who's trying to speak to me. Just unmute and say, ma'am, and I will respond accordingly. So with last week's lesson, we looked at a cell and the nucleus. Now, the nucleus is a very important part here because this is where DNA replication is going to take place. This is where we find our DNA. This is where protein synthesis is going to take place, mainly the first stage of protein synthesis, which is transcription. So it is important for us to understand the structure and functioning of the nucleus, which we covered. Looking from the outside going in, you'll have the nuclear membrane, which is filled up with the nucleoplasm, and then you have the nucleolus and the chromatin network, which will condense to form chromosomes. Now, in the nuclear membrane, there's spaces in between here, which we call the nuclear pores. These spaces over here, we call them nuclear pores. So we covered the DNA in detail, and then we looked at the functions of the nucleus. We then looked at the DNA and chromosomes, and I explained a little bit about genes. Then we looked at the nucleic acid. With nucleic acid, we have DNA and RNA. These are the two nucleic acids that we have. And I said you don't need to know the meaning of DNA and RNA. No one is going to ask you about it. Then we spoke about monomers of DNA. So in order for you to get DNA or RNA, you will need nucleotides. Now, those nucleotides will be grouped together in order to form a DNA or an RNA. Then we said that these are the locations for DNA and RNA. You find DNA in the nucleus, in the mitochondria, in the chloroplast. You find RNA in the nucleus and also in the cytoplasm. We also spoke about the history of DNA discovery, and we spoke about 4 scientists which were involved in basically researching and giving us information about the DNA structure. Then I skipped the slide. Then we said that nucleotides are made up of 3 parts. They're made up of a sugar, a phosphate group, and an organic base. So you'll have nucleotides for a DNA and also nucleotides for an RNA. Then in a DNA, the nucleotides, it's A, C, G, and T. The bases of a nucleotide. And then bases in an RNA molecule, that's A, C, G, and U. So we find U in RNA and not T. Then in terms of a DNA structure, we said DNA is double-stranded. So meaning it is made up of 2 strands. So these strands will be paired by the bases in the middle. Now A pairs with T and C pairs with G in those 2 strands, always. Now, the bonds that are responsible for base pairing are called weak hydrogen bonds. The word weak doesn't have to be there, so meaning the important word is hydrogen bonds. Then we spoke about the natural shape of DNA, which is double helix. So it's 2 strands that twist together and form a helix structure. Then we looked at the nucleotides, and then we did the calculations. In terms of the calculations, we said in a double-stranded piece of DNA, A will always be equals to T. So the number of bases that you have for adenine will always be equal to thymine, same with guanine and cytosine. And then we did the DNA calculations. Now, this is what we are going to start covering in this lesson, but just please give me 2 seconds. I can see the gate. People have been trying to call me. I'm not sure if there's any emergency. Please just give me a second. So I'm not sure if they're calling me for an emergency or what. Okay, if they call me again, please do excuse me, guys, for 3 seconds to just attend to them. Okay, in terms of RNA, remember RNA is the second nucleic acid. Now, RNA is a single-stranded molecule. In DNA, we spoke about 2 strands. And in these 2 strands, we are going to find bases that will pair with each other. Then in RNA, RNA is a single-stranded molecule, meaning it is made up of only 1 strand. Now, in this 1 strand, it will still have nucleotides that are bonded together to form this single-stranded molecule. Now, in the nucleotide, you will still find the phosphate group. You will still find the sugar, and in this case, the sugar is the ribose sugar. And then you will find a nitrogenous base. Now, you only have A, C, G, and U here. Now, it's important for us to note that as much as RNA is a single-stranded molecule, it does form a double strand with DNA at times. And what do we mean by this part? So during transcription, when we form proteins, RNA molecule will be paired with a DNA strand. When we form a messenger RNA, it will use a genetic code from DNA and actually pair with that genetic code. And when that happens, A will be paired with U and G will be paired with C. So RNA is single-stranded. However, at certain times, it does pair with DNA. And when that happens, A will be paired with U and G will be paired with C. That is important to note. Okay, so this is the single-stranded molecule for RNA, and we are able to see our nucleotides. This is 1 nucleotide. So that's the phosphate group. That is the ribose sugar. We can't say pentyl sugar here. We have to be specific. We're speaking about an RNA molecule. So that is a ribose sugar. Then a nitrogenous base. So in this particular diagram of a nucleotide, we are having adenine as the base, but in another nucleotide, we're having G. In another nucleotide, that is an A, then that is a G, and that is a U. We cannot be seeing T in an RNA structure at all. So you're able to see the different structures here. So that is your DNA. It's double-stranded in its helical form. And then you have RNA, which is single-stranded. Then we look at the 3 types of RNA molecules. So there's 3 types of RNA, and there's 2 which we are going to speak about in detail when we do transcription. The first type of RNA is messenger RNA. So for this type of RNA, you can write the full word of RNA, and then you can also opt to just write the letter M and then RNA in capital letters. Please make a note for this. If you decide to not write the full word messenger, you are allowed to write M, but your M needs to be in small letters, and then RNA needs to be in capital letters. So M needs to be in small letters and RNA has to be in capital letters. So if, for example, you write mRNA, messenger RNA, and you write M in capital letter and RNA also in capital letter, this will be marked wrong. We're not going to recognize that. Same if you write M in capital letter. Yeah, M in capital letter, and you write RNA in small letter. Let's say you write RNA. That's supposed to be RNA, but it's coming out as something else because of my pen. So M, I'm going to try again. Yes, RNA. So if you write something like this, we're not going to recognize it. So this is a type of RNA that is going to be formed inside the nucleus. So it'll be formed inside the nucleus during transcription. So it's going to use a genetic code from DNA and then deliver that genetic code to the ribosome. So I'm going to just say that and leave it as as is at this point, because then we're going to explain it in another lesson when we do protein synthesis. That's probably next week. Same with this one, transfer RNA. If you don't want to write the full word transfer, you can just write a small t and then RNA. So this is a clover-shaped molecule, and the function of this molecule is to carry specific amino acids. We are going to speak about this, what amino acids are, where we find them, and all of that. And that happens during protein synthesis. So when we form proteins, we are going to form proteins by actually forming a chain of amino acids. So amino acids are building blocks of the proteins, just like nucleotides are building blocks of DNA and RNA. Then we get ribosomal RNA. With ribosomal RNA, so this is the type of RNA that forms the structure of the ribosome with other proteins. So this one is involved in attaching the messenger RNA to the ribosome. So it will assist in translation here, which is the second stage of protein synthesis. So I'm just mentioning these types of RNA now, but they're actually going to be very important for us to understand when we do protein synthesis. Okay. Then, when we speak about a messenger RNA strand, we are going to form the strand using bases from a DNA, of which we will explain. But when we read bases on any strand, whether it's a DNA strand, a messenger RNA strand, transfer RNA strand, we normally read bases in threes. So in our diagram, you are able to see from left to right. So that is G, C, U. So that's how we also read them out loud in threes. G, C, U, A, C, G, G, A, G, C, U, U. Now, what you need to note, three bases in a messenger RNA strand, we call them a codon. So these three bases here are called a codon, just like these three bases here, just like these three bases here. Hence, we are counting them as codon 1, codon 2, codon 3, codon 4, codon 5. So there are 7 codons here. If someone were to ask you how many bases can we see on this strand, obviously you will say 7 multiplied by 3, or you can just physically count the bases. So codon, 3 bases in a messenger RNA strand. Then when we look at a transfer RNA strand, please do give me a second. I can see the gate is trying to call me again. I'm not sure if there's an emergency. Please just give me a second. Okay, I'm back. Sorry about that, guys. Okay, so with transfer RNA, this is a type of an RNA molecule that is important in carrying amino acid. Please familiarize yourself with how the transfer RNA looks. This is very important. I'm just double checking if I unmuted. Okay, cool, that's fine. At the bottom of the transfer RNA, we are going to find 3 bases. And these 3 bases that we find at the bottom of our RNA molecule, we are going to call them anticodon. Now, these 3 bases, which we call anticodon, will need to be complementing the codon of the messenger RNA. Remember, 3 bases in a messenger RNA, we call them a codon. And then 3 bases in a transfer RNA, we call them an anticodon. This anticodon, 3 bases, they are going to complement bases of the codon. So on one side, the transfer RNA will carry the anticodons, and then on the other side, it will carry a specific amino acid. Now, this is a building block that is available in the cytoplasm. And again, I will explain it in detail in protein synthesis. So the amino acid that will be carried on this side of the transfer RNA will be determined by the anticodon, meaning the bases here at the bottom. So it will not carry just any amino acid. And I know there is a common question about the names of the amino acid. Do we need to know them? You don't need to cram them or know them or anything like that. They will give you a table of the different amino acids that we have. So you don't need to cram anything. Then we look at the differences between DNA and RNA. We've covered everything about DNA now, and then we covered everything about RNA. Now, we need to look at the differences, and they like asking this question. So by just looking at the diagrams, I feel like me and you can answer without even reading the notes. So if you look at the structure of DNA, you can see that it is, made up of two strands. There is a strand here and there is another strand. So DNA is double-stranded. Then when we look at RNA, we are only seeing one strand. RNA is single-stranded. We've compared the first characteristic. We can also look at something else. We can look at the bases. Now, in DNA and RNA, we have bases A, C, and G. They are similar, right? The only difference is T and U. So DNA will contain base T, which is thymine or thymine, and RNA will contain base U. That's the second difference. Another difference that we can speak about is the pairing of bases. We can see that in DNA, we are finding bases that are paired. A is paired with T, G is paired with C. Then in RNA, the bases are not paired because this is a single-stranded molecule. Any pairing that we speak about in RNA will be pairing that occurs during transcription. So that is important to note. Now, this is a nice table that differentiates between DNA and RNA. I will have a lesson, I don't know when, maybe May or June, for you guys to address skills, rules that you need to follow when you are asked to draw a table, when you are asked to draw graphs, how to answer scientific investigations, how to draw bar graph, line graph, how to get investigations and, well, variables, reliability, validity, all of that business. But when you are asked to draw a table, I'm going to mention this because they normally do ask you. Make sure that you draw all your table lines. What do I mean by this? This is your table lines. And then you make sure that you separate information for the two things that you are comparing. In this case, we are comparing between DNA and RNA. Now, one important thing is the first row. This is a row. We read a row from left to right. The first row should have your column headings. And you are telling us, oh, okay, in this column is top to bottom. So in this column here, I'm going to speak about DNA. And in this column here, I'm going to speak about RNA. So you write it down. Your table needs to have a heading. So if you look at our notes, we do have something that says tabulate differences between DNA and RNA. That is not the heading of the table. The heading of the table will say a table. You are telling us this is a table showing us differences between DNA and RNA. Fun fact, by writing a heading and drawing a table before you even give an answer, it gives you 1 mark. By just writing a heading of the table and writing the table lines. Now, another thing that you need to take note of when you are comparing between two things, in this case, we are comparing between DNA and RNA. You do not use one-word answers. You're not going to be marked. You do not use one-word answers at all. You give us a sentence that makes sense. So what do I mean by this? If, for example, yes, now we are comparing between DNA and RNA, and we say DNA is a double-stranded molecule, we are telling you in terms of structure how DNA is. And then RNA is a single-stranded molecule. You get a mark for that. But If you decide to just write under DNA, you write double. Then under RNA, you write single. Those are one-word answers. We can't read. What do you mean double? Double what? Single what? So we're not going to mark that. So please do write sentences that make sense. Then in DNA, there's also pairing of bases. In RNA, no pairing of bases. DNA contains nitrogenous base thymine. RNA contains nitrogenous base uracil. Check how we wrote the whole sentence. You can't just say in DNA, you write thymine. In RNA, you write uracil. What about them? We're not going to give you a mark for it. Then for the sugar, we can also compare the sugar. DNA contains deoxyribose sugar and RNA contains ribose sugar. Then you can speak about the length of the molecule. So the length of the molecule, DNA is a longer molecule and RNA is a shorter molecule. I'm not a fan of these last two, so maybe do try and avoid them. The other one speaks about the location. The other one speaks about one molecule being coiled and another not being coiled. I'm not a fan. So when you are asked to differentiate, please focus on the first four. Now, you can also write these words on the side so that you remember the characteristic that is being compared here. Because in the same row, you need to compare the same characteristic. What do I mean? If you check on this row, we are comparing the sugar. So we speak about the sugar in DNA and speak about the sugar in RNA. We don't speak about the sugar here and then speak about a shorter molecule there. So that means this side, we are speaking about the sugar, and the other side, we're speaking about the length. We're going to penalize you. So if you check on this row, we are speaking about the length. And then in this row, we are speaking about the comparison of the base. And then in this row, that's the pairing of bases. And in this row, the structure or the strengths of the molecule. I hope that makes sense. Remember to stop me anytime, guys, so that I can address whatever question that you have. All right, but we have something cute now on our screen. So I'll give you 2 minutes for you to try and answer this. Okay, 2 minutes is not enough. 3 minutes. So it's 18:30 now. At 33, we are marking. Please be ready to be called. I will call people's names and then we answer. So this is simple, simple, simple stuff. There's 2 more minutes. Just get water. There's 1 more minute left, guys. Please, when I call your name, do unmute and answer for us to avoid wasting time. Even if you're not sure whether your answer is correct or not, this is a lesson. We are learning. So I don't just want perfect answers. I need us to obviously be open to learning and to be corrected, because at the end of the day, we want to get good marks. So I think it's few more seconds left. Okay. Can we start marking? Thumbs up? Or do you need 1 more minute? Let me go back to you guys. Can we start marking or you need a minute? 1 more. You can thumbs up or you can unmute and say, ma'am, 1 more minute, something. Okay, I'm seeing a few thumbs up. All right. Thank you. The diagram below shows a nucleic acid molecule. So this is a nice, simple question just to check our understanding about DNA and RNA. The first question says, identify the molecule. They've said that it's a diagram showing a DNA, showing a nucleic acid. Okay. Ayabonga, please identify the molecule.
DNA molecule.
Thank you so much. DNA molecule. If my pen doesn't want to write or it messes up, guys, please do just listen to what we are saying. Remember, you can or you will be marked, not even can, you will be marked correct if you write DNA. You don't need to now remember the meaning of DNA, deoxyribonucleic acid, because you're going to mess up the spelling and then we penalize you. Then provide labels for 1, 2, and 3. Let's see who's going to give us 1. 1, Sibabalo, please do give us 1.
Is it adenine?
Adenine, that is correct. That is definitely adenine. Okay. If anyone needs to ask something, please do ask anytime. If maybe, for example, you wrote C or G there by 1 and you're trying to figure out why are we saying it's A, please do ask any second and I will definitely answer. So it is adenine. Then 2, Nklamulo. Yes, I'm hearing someone. Oh, okay. Nklamulo, can you give us 2, please?
Ma'am, is it phosphate group?
Phosphate group. Let us look, Nklamulo. So in a nucleotide, we find 3 parts. So this is the nucleotide. So there's this part here and this part here that has 5 sides and then a base. So definitely this is a base, which will be a T. So between these 3, which one is a phosphate group? Is it this one? I'm going to label this one X. Is it X or 2, which is a phosphate group?
It's 2.
X.
So X will be a phosphate group. And 2, anyone please do help us out. What will 2 be?
Deoxyribose sugar.
Deoxyribose sugar. So the shape that has 5 sides. Remember we say this is a pentose sugar. Pentose means 5. So when we look at a nucleotide, the circle is the phosphate group. And then the one with 5 sides will be the sugar and then bonded to a base. So the one that they've asked us in this question is this one, which is a sugar. So deoxyribose sugar. Can I check? Is there anyone who wrote pentose sugar? You can raise your hand. I should be able to see people who are raising hands here. Is there anyone who wrote pentose sugar? Okay, I'm seeing a Julian. Anyone else? Oh, wait, quite a few. For this particular question, if you wrote pentose sugar, you're going to be marked wrong. And why is that? You are able to see that the molecule on this particular diagram is a DNA molecule. You need to specify the sugar that we find in a DNA molecule, which is deoxyribose sugar. So we can't just say pentose sugar because we have pentose sugar in both RNA and DNA. But for our specific diagram, this is a deoxyribose sugar because we find it in DNA. So I'm happy for us to make mistakes on the lesson because when we make mistakes here, we're not going to make mistakes in a test at school or in our final exams. So please be specific when you are asked about the type of sugar. So if it's in an RNA molecule, you will say ribose sugar. If it's in a DNA molecule, you say deoxyribose sugar. I hope that is clear. Okay, then the last pentose sugar. So pentose sugar, I'm seeing a comment here from Akona. Pentose sugar is a sugar for RNA. For DNA molecule, which is the molecule that we have on the board, the sugar is DNA. Maybe let me teach you guys some cramming skills. If maybe we forget this cramming skill, please do apply it. So DNA starts with the letter D. The name of the sugar is deoxyribose sugar. Ribose sugar. DNA molecule. The name of the sugar, deoxyribose sugar. So both start with the letter D. Then RNA molecule. The name of the sugar, ribose sugar. RNA starts with the letter R. The type of the sugar we find in RNA ribose starts with the letter R. Okay, so I want us to remember that we can never mess it up after today. Then 3, give the number. Yes, I'm hearing someone. Is someone speaking, Cindy? So?
Yes, sorry to take you back on question 2 at labeling 1. What if instead of writing adenine, I wrote nitrogenous base?
Okay, good question. So the question, let me change my color. So you are asking here on this question where we said provide labels for 1, and there is 1 here. So then the understanding is that section is section of a nitrogenous base, correct in that understanding. But we needed you to tell us which base is found there. So this is the old school way of figuring it out. So where would you go? You would be led by T and look at the shape of T. And obviously T is going to be paired with A. And then you match the shape for A with this one. So that's how you would have figured out. So in this case, again, you couldn't just say base. Which base is it at this particular point? That base was adenine. So you're going to be penalized, actually marked wrong for that. So good question also. All right. Is it fine, Cindy?
Thank you, ma'am.
Okay, thank you so much. Then I need
Yes, ma'am.
someone who's, yes, I'm listening. Lebu, please do answer question 3 for us. Lebu Khang, give the number of nucleotides that are shown in the diagram. How many?
Ma'am, I think there's 10 nucleotides.
You think there is 10 nucleotides. And
Yes.
I also think there is 10 nucleotides. So
Ma'am,
there
question.
is, yes.
So, right there by adenine and thymine, right?
Over here?
Yes, at the top. When they're together, what do we call that? Is that a nitrogenous base?
What's together? A and T?
Yes.
A base pair.
Oh, okay.
So it's a nitrogenous base. When you are looking at it on its own here, look at this diagram on the right. So this is a nitrogenous base that will be there. And the nitrogenous base there can either be A, T, C, or G. But if you are looking at a T being paired with an A or a C being paired with a G, they are called the base pair and they are called the base pair.
Thank you, ma'am.
All right. So definitely we do have 10 nucleotides. We can count them 1, 2, 3, 4, 5. My pen is writing and they're not writing, but I'm sure you guys can see what is happening here. Then it's 5 this side. Obviously, it'll be 5 the other side. We didn't even have to count them like that because if we count the bases, each nucleotide will contain 1 base. So if you count the bases, it will give you the number of the nucleotides. Stop me any second, guys. Please remember to stop me any second. Then there is this one. So I'm also going to be nice and give you 3 minutes. It's 1842. At 1845, we are marking. Oopsie, my laptop is low. Please try your best, and we don't leave empty spaces at all, And the teacher gets really frustrated when learners leave empty spaces, because an empty space means I am not even trying. I don't want to try. Here, we are going to try our best. We're not leaving any empty space. We try our best. This is a platform for us to learn so that we can be very confident going into a test or an exam. So we are going to use this platform for us to learn and perfect our answering skills. Oh, I yep too much. 2 minutes left. I'm going to be quiet. Okay, are you ready for us to start marking, guys? If you need 1 extra minute, please just unmute and say 1 more minute, ma'am, or 2 more minutes. I don't want to rush anyone.
1 more minute, ma'am.
Thank you. I'll give you 1 more. I think that was 082. 082, are you sorted now? Okay, I think we can start marking, guys. Thumbs up if we can start marking. Oh, perfect. I'm seeing a few. Perfect. All right. Figure 1.4. This is the figure that we are looking at. Represents a part of a nucleic acid molecule. Study the diagram and answer the questions that follow. Identify the nucleic acid. Tato, please do identify it for us.
I said DNA.
Definitely DNA. So the nucleic acid is DNA. Question 5. Provide a reason for your answer. Why are we saying this is a DNA molecule? Oratile, this one is for you. Give us an answer. Oratile, please give us a reason why we're saying this is a DNA molecule. Okay. Akona, please do give us a reason. Okay, if you're ready to answer, let me just ask people to raise hands then. Please do raise your hand if you're ready to answer, guys, so that I can call out your name. Okay, Julian.
Okay, so there are two strands that are used as a template and they're connected by the hydrogen bond.
Perfect. So we are seeing two strands, that's a tick. We are seeing the presence of hydrogen bonds between base pairing, that's a tick. Does anyone else have another reason that they want to check? You can just unmute and give me your answer, whatever answer that we didn't mention. So you can check if it's correct or not.
I said it contains the base adenine and pairs with cytosine, and it shows as a double-stranded molecule.
Okay, just read your answer for me again, please.
It contains the base adenine and pairs with cytosine, and it shows as a double-stranded molecule.
Okay, the first part of the answer would be considered incorrect. The last part of the answer, I will give it to you. You spoke about a double-stranded molecule, meaning you're telling us there are two strands. But the first part of the answer, you're saying it has adenine, which pairs with cytosine. So also RNA has adenine, so we can't consider that as correct. And then you said A, which is adenine, pairs with C. That is technically also incorrect because A pairs with T. So I want us to look at that. Let me just see who was answering. Please, let's look at that. So the last part of your answer, double-stranded, that is fine. But you mentioning A or mentioning C or mentioning G, that would be incorrect for this particular answer. Actually, A doesn't pair with C. Okay, is that fine?
Yes.
Okay, thank you so much. All right. I am happy when I hear these responses, guys. And if your answer is incorrect, may that please not affect your confidence in any way. Like I'm saying, this is a platform for us to learn. So I'm not in class where I can walk around and see your answers. This is online. So once you say something and I need to rectify it, I'm rectifying it so that you don't make that mistake in a test. You go there and get good marks. Okay. Then let's label the following part 1. There is part 1 here, and they want us, let's label part 1 first. If you're ready to answer these questions, please do raise your hand so that it makes it easier for me. Inganati, part 1.
Part one is a phosphate ion.
It's a phosphate. That is correct. Phosphate.
Ma'am,
Yes.
ma'am, can I ask, am I wrong if I said it is a phosphate group?
Phosphate group is fine. So whichever word you added that is technically accepted in science after phosphate, that is fine because the tick will be at phosphate. That's what we are looking at. So if you said
Okay.
phosphate ion, I don't know, a phosphate group, I can't remember what other options are, but what we are looking for is phosphate. So you're not going to be penalized if you say phosphate group. Okay, then please label the nitrogenous bases 4, 5, and 6. There is 4, and there is 5, and there is 6. Please raise your hand so that you can give us all 3. Specifically,
I said 4 is guanine, 5 is guanine, and 6 is thymine.
okay, let's figure this out together. So you are saying 4 is what?
Guanine,
So that's G. 5 is
G also
G also, and then 6,
is T.
it's T. All right, do you guys agree with these bases?
No.
Okay, which one are you
No.
not agreeing with?
The,
Number 4.
the
first
number
one.
4. The fourth one.
Number 4.
Number 4.
Okay, so let's start with 6. Are we happy with 6?
Yes.
Definitely
Yes.
happy with 6. This is a T. Let's follow the shapes also here. Let the shapes and the bases guide us. So if we are having this shape going in for A and this one having a space for A to fit in, this will be T. We are happy. And for this one, we are having C with this curvy shape, which will be bonded by this particular shape. Also at the base, there will be G. But if we look at this shape, Let's match it with other shapes that we've looked at already. This 3 looks exactly like the 6, and we said 6 is a T, so that means here at 3 there'll be a T. So meaning our base at 4 will be which base? Will be A.
Adenine.
Thank you.
Adenine.
Thank you. I'm so happy, guys. Thank you so much. Right. So wherever we make mistakes, we correct them in this lesson so that we can go to that test and exam with confidence. So 6, let's label, 7, sorry. What is the collective name? I need to see your hands. What is the collective name for the parts numbered 1, 2, and 3? Mohau?
Oh, sorry, I got a little bit of distracted. We are at question 7, right?
Question 7, yes.
Oh, I wrote nucleotides.
Perfect. Nucleotide. Is there anyone else with a different answer that wants to check if their answer is correct or not? Nucleotide it is. All right. Then, so 7, we are done. What will the instructions coded on this molecule be used to make? I want to hear this one. What is the what will the instructions coded on this molecule be used to make? I need to see the hands. Okuhle?
I said proteins.
Protein. That is perfect. Definitely protein. So the protein, yes.
Ma'am,
Can
why
you
do you
please explain the
Okay, let me see who's speaking first. The lady that was speaking, you can ask first, then it will be the gentleman.
Can you please explain the question?
Okay, noted. Then can I hear the second question?
I was going to ask, what if I wrote DNA replication?
DNA replication would be incorrect.
Okay.
So let me explain the question. So what will the instructions coded on this molecule be used to make? Now, there is a section that we are going to cover next week, which we call protein synthesis. We make proteins, but these proteins are actually made from a DNA code. So what is our molecule here? DNA. And in this DNA, we get a code that is going to make the proteins. The question says, what will the instructions coded on this molecule be used to make? We make proteins by using a DNA code. So we are going to cover it next week. So we can't say DNA replication. We are going to cover DNA replication just now. In DNA replication, we're basically using DNA to make another copy of DNA. So it's DNA making a copy of itself. If I have a page over here, and I make a copy of that page, I will have 2 pages that look exactly the same. So that is DNA replication. So very different to protein synthesis, but easily confused. All right, I hope that makes sense. What is the natural shape of this molecule? I can see those other hands that are not going up at all. Other people are not raising up their hands. Even if you're not raising your hand, I'm still going to ask you to answer, please. So please do raise your hand. Mihle, please do give me an answer for this one. What is the natural shape of this molecule? Okay, let me go back to people whose hands are up. Sigelela, what did you say? Okay, anyone, guys? Anyone? Let's answer
Yes,
so that we can move.
yeah. Can I try, ma'am?
I'm listening.
Okay, I said double helix.
Double helix. Let me put 2 options here on the board.
Okay.
So we're going to check how many people said double helix. And then check how many people said double stranded. Double stranded. Okay, I'm going to check. Please raise your hand if you said double helix. So I'm going to check here If you said double helix, please do raise your hand. Even the people that don't want to answer, I'm not going to ask you to answer now. I'm just trying to figure out what's happening. So those that said double helix, okay, I can see a few hands. All right, please do remove your hands. Well, that's just clicking. Please do take your hands down. Okay, I need to now see people that said the shape of the DNA is double-stranded. Please do raise a hand if you said double-stranded. Double-stranded. Please do raise your hand. Okay, I can still see that there's a few people. The correct answer is double helix. If you wrote double-stranded, you will be marked wrong. Why is that? They are asking you the natural shape of DNA. Yes, DNA is double-stranded. That is correct. What do we mean by double-stranded? We mean that DNA has two strands. This is what double-stranded means. It is made up of two strands. But that's not the natural shape. What happens to these two strands? They actually twist. That is the natural shape. So yes, it's two strands that intertwine. So if we say double-stranded, you're not going to get a mark. You need to say the natural shape is a double helix shape. That is very important. We are addressing everything in this lesson so that we can get good marks. I'm here now, DNA replication. Before I start, is there any question about anything that we've covered so far in this lesson? I've been speaking for a whole hour. Anything that you need clarity on, which I've just covered, or any of the questions from the two activities that we did, I'm happy to answer before I move to DNA replication.
Ma'am,
Yes.
going back to what you just said, if we say double, what did I write? Double-stranded helix or double-stranded helix, would that still be incorrect?
I'm trying to remember what they said 1 year. Double-stranded helix. For this lesson, I will say wrong.
Okay.
Because you know why I'm saying wrong, Lebu?
Yes, ma'am.
Because I want you to remember that it's just double helix for a test.
Okay.
So double-stranded helix, yes, it depends what we are arguing about at the marking center, because someone can say, let's ignore the word stranded. The learner has the two words that we want. They have the double and the helix, let's give them a mark. But then someone else will say, this learner is confused. They're speaking about double-stranded helix. They're adding words that are not supposed to be there. So we don't know what the mood will be in this year's marking. So that's why I'm going to say for our lesson, let's say that is wrong, so that we can learn when we prepare for a test that the shape is just double helix.
Thank
to be
you.
safe. All right. Anyone else with a question? Because I'm about to just push for a good 30 minutes teaching this without taking any questions.
I
All
want to
right.
know.
Okay, cool. We're gonna do DNA replication. If you have a page with you, I'm gonna plead that you use that page and we go through this process together. It doesn't have to be a big page, just an A4 or just a half size of an A4. I think it's an A5 or whatever. And then we draw the diagram together. So I'm going to look at DNA replication in the notes and then look at DNA replication in the diagram, how exactly it occurs. This is very important. And next week, when we do transcription, we are going to again look at DNA replication because protein synthesis is first stage and DNA replication, they look very similar because they take place in the same location. So let's get the basics straight and hopefully you look for a paper so that you can draw the diagram once I start drawing the diagram. So let's break down this process. There is a 99.99999% chance that you will be asked about this. So DNA is a nucleic acid that we've already spoken about. And we said that this DNA we find in the nucleus. So this will be our cell. And then in our cell, don't move. I'm not sure why my slides are moving. Then in this cell, there's other organelles in the cytoplasm. But one of the organelles that we find in the cytoplasm is this large organelle, which we call the nucleus. Now, in the nucleus, there's other structures, but then we have the chromatin network. And this is a network that contains our DNA, which is information that makes you. The reason you have a certain skin tone, the reason you have a certain color for your eyes, the reason you have a certain color or texture for your hair, your blood group, and so many other things, which are body characteristics. It has nothing to do with behavior per se, because behavior it can be influenced by external factors. So it has nothing to do with how people behave. Now, the word replication means to replicate or to copy. So in this process, we are going to make a copy of DNA, hence DNA replication. Now, we need to find out where exactly this process is going to take place. Where is this DNA replication take place? And when exactly does it take place? Why does it need to take place? What's the significance or importance of this process? And how exactly it takes place? So we are making a replica or copy of DNA, meaning if this is our DNA, which is the chromatin network, we are planning to make another DNA, which is exactly the same. I made an example and I said, if I have a test, my one-page test here, and I put it through a photocopy machine, that page will come out the exact same. If it has 7 questions, there will be 7 questions. So I would have made a replica of the question paper. So we've answered what the process is. We are producing an identical copy of the DNA of a cell. Now, what will make this DNA identical? It's actually the sequence of bases. The reason me and you look different, it's not because I have certain bases that you don't have. Me and you have ACTG, and ACTG you also have. The only reason we look different is the sequence of my bases will obviously be different to your sequence of the bases. So in the DNA replication, we are going to look at the sequence of bases. So if we say the DNA is identical, this DNA's bases sequence should be also identical to this DNA's bases sequence. That's all that we're looking at. Where exactly will this process take place? It will take place in the nucleus, hence my diagram over here. When will it take place? Okay, we know what it is. We are making a copy. We know where it's going to take place in the nucleus, but now when exactly? There's what we call a cell cycle. A cell undergoes cell division. But before it undergoes cell division, there's different stages that it will need to go through. One of those stages or phases is called interphase. So just before cell division, and there's 2 types of cell divisions, you can make a note here. There is mitosis and meiosis. Now, when we speak about the cell division, we are speaking about a cell physically dividing to form other cells. Now, the reason DNA replication occurs is so that the genetic material becomes doubled. So if this genetic material is doubled, when the cell divides and makes 2 cells, meaning we are going to have a cell here and another cell here, each cell will end up having genetic material. Now, the genetic material for the cells at the end of cell division, particularly mitosis, the cells will have the same genetic material. It becomes different in meiosis, which we will address at a later stage. So
Yeah.
what is it? Yes, I'm listening.
I wanted to ask, if I was to be asked when does DNA replication happen? If I were to write during interphase, during, if I were to write, it happens during interphase before cell division without mentioning the part of cell cycle. Will I get a wrong or a right mark? Without mentioning the part that says cell cycle.
All right, good question, Mohau. When they ask you, when does DNA replication happen? The only answer that you need to write is interphase. That's it. Nothing else.
Okay.
So of the cell cycle before division, you don't need this whole part. Once you say interphase, it's a tick. That's all we need. Same with where. You just write nucleus. Then that's it. I hope I've answered your question, Mohau.
Yes, yes. Thank you.
Okay, thank you so much. Thank you, guys. Remember, feel free to stop me anytime. So what is it? We are making a copy. Where does it take place? In the nucleus. When? Interphase. So interphase is the phase whereby the cell will make a copy of that DNA, and then cell division starts. Mitosis either starts or meiosis starts. Now, why does this process need to take place? We need to double the genetic material. Why are we doubling the genetic material? So that it can be shared between the resulting daughter cells. We are forming daughter cells here. So every cell needs a genetic material. There is no cell without genetic material. So in order for each cell that is formed to have genetic material, we are going to make copies so that they can be shared between the cells. How does this whole process take place? What it is? Copy. Where? Nucleus. When? Interphase. Why? We're doubling the genetic material, and this will result in identical daughter cells during mitosis that we are going to address at a later stage. How exactly does it take place? We are going to skip that and we find out how. This is very important. They will ask you to describe DNA replication. And I can assure you, when you are asked to describe DNA replication, they need you to mention these points that I have on the slides. And can you guess how many marks this process will be allocated for? 6 marks. We are not going to leave empty spaces, and we are not going to lose marks in DNA replication. Because once we understand and visualize the process, we should be able to write down the points. When they expect you to describe, you're not going to collect information from the atmosphere. You have the information on your slides. Now I'm going to go through these slides first, and then I'm going to draw the diagram in order to explain DNA replication. Now I need you to visualize this happening. Now, when you are asked to describe, it's 6 marks. Please make a note so that when you study this, you can study it with the intention of knowing 6 points for 6 marks. Now, what I also need you to know here, as much as you have the numbers, number 1 to 9 in our slides for the points. Fun fact, in an exam, if you were to write these points and include the numbers, you will be penalized. Don't include the numbers. You can include dashes or dots, and then you write another dot, and then you write your points. Do not write numbers. Also, when you are studying for the 6 marks, please make sure that the order of the points is correct. Remember, if I were to ask you now, I always say this in my groups, I love food. I can cook what I want to eat, but I can't bake. But if I were to ask someone, okay, please guide me, I want to bake scones. So what should I do? When you give me the directions of how to make scones, you're going to make sure that you give me information step by step. What's the first thing that I need to do? And then this, and then step 3, and then step 4. You're going to tell me to mix the flour with water, with milk. I hope that's what you mix, and with eggs. I've seen people do that. But if I were to ask you to tell me how to bake scones, you're not going to say, put the mixture inside the oven and then take it out, put water and eggs. You are messing up the order. If you mess up the order, you lose the marks. So I hope that is clear. All right, let's look at this. If you have these notes, please make note of everything I'm going to say. If you have not printed them, it's not a crime. Please make note somewhere because these points are important. This process, we remind ourselves, it happens during interphase and it happens inside the nucleus. Now, remember, the natural shape of the nucleus is that it is double helix, meaning it's two strands that are twisted together. Now, when this process occurs, we need those two strands to unwind. But how do we phrase it? The DNA double helix unwinds. There is a tick. I need those 4 words to give you a mark. If there is a word that is missing, I don't give you a mark. That is our rule. That should be the rule at school. And that is the rule at the end of November at the marking center. If you don't mention the word DNA, you just say double helix unwinds, wrong. If you say DNA double-stranded unwinds, wrong. I need these 4 words for you to get that 1 mark. So that's what I'm saying. You need to visualize it or picture it. This is the natural shape of the molecule. So the first thing that needs to happen to this DNA double helix natural shape, it will need to unwind. That is the first mark. Then when the molecule unwinds, you get the 2 strands. You then get those 2 strands, which are still paired together because there's bases in between there, which are paired by the hydrogen bonds. Now, our goal is to have these molecules separated. But the first thing that needs to happen, in order for the molecules to be separated, we need to break the bonds that have joined the base pairs. That is the second point. So what do I need here to give you a mark? Hydrogen bonds between the 2 strands break. Tick. I didn't mention weak. So I'm going to put weak in bracket because the word weak is not needed there. Whether you have it or not, it doesn't change your answer. What are we explaining? The bonds between the 2 strands are going to break. Whatever that is joining the strand breaks. Now, when the bonds break, what happens to the molecule? It actually unzips. So it will have this shape over here. So you can see, I think we've all worn a trouser or a jean or something like that. When you unzip it, you get that kind of a shape. So that's what happens. Now, this sometimes gets a mark and sometimes doesn't get a mark. So please also highlight it on your side. You can get a mark by just saying the molecule unzips. Now, this is technical marking. I am teaching you a skill that is going to allow you to get 2 or 3 marks by just having 2 points. Please note this. These 2 words are not friends. These 2, unzips and unwinds, they are not friends. And you are not going to swap them around. You're not going to say DNA double helix unzips, wrong. So these 2 words, not friends, not siblings. We're not going to find them substituting the other. They are not twins also. We're not going to find them swapping around at all. Now, please also note, the order is important. You can't say the molecule unzips, the hydrogen bonds break, and the double helix unwinds. You've lost all 3 marks. The molecule cannot unzip before it unwinds. So that will be wrong. So we've covered the first 2 points. So what did I say? The DNA double helix unwinds, and then the weak hydrogen bonds between the 2 strands are going to break, and the molecule unzips. Visualize it. Now, when the molecule unzips, we then end up having these 2 separate strands, which is what we want. Yay, we are on point number 3. But now what's going to happen to these 2 strands? We are going to use them. So the words that you need to use here, as much as this is not highlighted, I need you to highlight it. We have 2 strands. Both of these strands are going to be used to serve as a template to form a new strand. The tick is normally at the end. So these need to be underlined or highlighted. That whole sentence, I need each and every word. Now, if you don't want to use the word each, you can use the word both. Both strands are going to serve as a template to form a new strand. What do we mean by serving as a template? When something serves as a template, you're going to use it to make something else. So we are going to use these two strands, original strands, to make new strands. That is point number 3. Then point number 4, since this is happening in the nucleus, in the nucleus we have DNA nucleotides. Yes, we'll have the DNA itself in a double helix form, in a chromatin network or in a chromosome. And then we have free DNA nucleotide. What do we mean by this? Remember, when we speak of a nucleotide, we're speaking about that phosphate, which is bonded to the sugar, deoxyribose sugar, and the base. And this base can be A, T, C, or G. These DNA nucleotides, where are we finding them? We are finding them in the nucleoplasm. They are going to attach themselves to the exposed bases of the two original strands to form a complementary strand. How are they going to attach themselves? A is going to be paired with T and G is going to be paired with C. It will make more sense when we do the diagram. Now, the two original strands that we have that act as a template to form a complementary strand. At the end, we are going to have two genetically identical DNA molecules. But now each molecule will consist of one new strand and one original strand. Now let's figure out how this whole thing happens. Yes?
But can I please ask a question? At number 4, if we don't, like, are we going to get penalized if we don't add the nitrogen bases like A to T and C is to G?
No, you won't be penalized. The tick is normally here. So it will be, this is what is important, DNA nucleotides in the nucleoplasm attached with complementary nitrogen bases. The tick is normally there. So you can add A to T, G to C, because sometimes it gets a separate mark. So you can add it.
Thank you, ma'am.
Okay, thank you so much. Okay, now we are going to our, is there another question?
Yes. I wanted to ask if I have my DNA replication on my textbook is different from this one. Can I still get marks from it? Because for my textbook, it doesn't end like this one.
How does yours end?
Mine says at the end, it says each molecule consists of one original strand and one new strand.
There you have it. New molecule consists of one new strand and one original strand.
Yes.
Yeah. So the information in the textbook is correct. It will be the same. But like I said, our strands, not strands, our notes, we update them. So these are straightforward. So what you've said is the same as this. You're not going to be penalized. You said each new molecule, there is a new strand and an original strand. So that'll be fine.
Okay. It doesn't include the process is controlled by enzymes. There's no part in my textbook.
Okay, good one. There is no mark for this.
Okay.
There is no mark for this. Point number 7, there's no mark for it also. So everything else there is a mark for, but point number 9 and point number 7, there's no mark for it.
Ma'am, can I ask?
Yes?
Ma'am, what is the difference between point 3 and point number 5?
Point number 3 and point number 5. Point number 3, we are still having our two strands. And I think when I do the diagram, it will all make sense. We are having the two strands and we're saying they are going to act as a template for us to form new strands. Now at point number 5, the original strands will act as a template to form the new complementary. I think they speak about the same thing. Let me just figure it out quickly and remember how we did this. The original string serves as a template, okay, to form a new string. The original string acts as a template to form. Okay, it's 6 and 9. It's the same thing. It is the same thing here because it's the original strand. That is still the same thing. Unless I remember something specific that was discussed in the meeting, then I will say it. I'm not remembering anything specific at this point. If I do remember, I will mention. All right. Is there any other
So,
question?
yes. So, ma'am,
Okay.
will you be penalized if you will not write point 3?
If let's say you substitute the 0.5 with point number 3, if it still means the same thing, then you should be fine.
Okay, thank you.
There's no way that, let me figure it out now, each, so that's fine. Okay, I'll figure it out now. Let me think of something, I'll figure it out. Because I don't want us to substitute, I don't want us to say, let's substitute it, then you end up being penalized. I think when I do the diagram, I'll remember something, then I'll tell you. Okay, so I'm going to share a Word document that is going to be blank. So this is when you take out your paper. There won't be anything on your screen for now. Let me see what's happening. Okay, so just give me 5 seconds. Please just give me a few seconds. I'm trying to get this Word document. Okay, that's done. So once you open Teams and record and you add a device and you add a lot of things, the laptop becomes slow because it's using a lot of things at the same time. Okay, it's coming. Okay, it should be fine now. Okay, it's on my side, but it is still showing me that it's not on your side. So, okay, there it is. Perfecto, 5 seconds. It should be on your side. 5, 4, 3, still not on. 2, 1. Wait. Okay. Let me figure out what's happening. Okay, please bear with me because it's not giving me an option to select that. Okay, then here. Okay. Should be fine. So I've closed everything. Wow, it should be on your side now. It should be thumbs up if you can see it. It's a blank Word document. Okay, there's nothing so far. So there's no one reacting to anything. Oh, please thumbs up if you can see it, guys. It should be on your side. It's telling me it's on your side. Okay, cool. Oh, okay, cool. Sorry about that, but I can't control how slow the laptop is. So yeah, thank you so much. Let's start. So we are doing DNA replication and now I'm running out of time. So this is what we're going to have. DNA replication. Okay, so what is the main goal here? Our main goal is to make a copy of DNA. And where is this whole process taking place in terms of location? It is taking place in the nucleus. When exactly is it taking place? It will take place during interphase. So that is very important. So I'm going to draw a diagram of the nucleus, and please, please, please do the same thing that I'm doing on my side. So remember, the nucleus is found within a cell. So if maybe, let me be nice and start by drawing the cell. Let's see if I can have space here and start by drawing a cell. And if we are speaking about, ignore this part, please, because if I erase it now, it's going to erase the whole thing. So this is the cell. If we are obviously speaking about an animal cell, this would be a cell membrane. And filling up the cell membrane will be that jelly-like substance, which is the cytoplasm. I'm mentioning these words because they are important. Then in the cytoplasm, we find other organelles. And one of the organelles that we find in the cytoplasm is the largest organelle, which is the nucleus. There is the nucleus. Making up the nucleus is the nuclear membrane. Nuclear membrane. Oh, my pen is spot on today. Okay, then filling up the nuclear membrane, we remind ourselves, filling up the nuclear membrane, it is the nucleolus. It can be. It can be. It can be the nucleolus. Nucleoplasm. Thank you. Okay, so we remind ourselves now we're going to start. So I need you to remember all of those points. So the first point, we said the DNA double helix unwinds. I need those 4 words. First, mark. Now, this is the DNA double helix. It's going to unwind. I'm just allowing you to picture it. Then the weak hydrogen bonds are going to break between the 2 strands and the molecule unzips. That was point number 2. We can't show all of that now. Then, when the molecule unzips, it will form 2 separate strands, which we can show. I'm going to try and change my color. We can show the 2 strands that are going to be formed. These are the 2 strands that will be formed. DNA double helix unwinds, weak hydrogen bonds break, and the molecule unzips. We then end up having 2 separate strands. When these strands are now separated, we will be able to identify the bases. My lines are skewed. Your lines don't have to be skewed. They can be straight. It will still work out fine. So I'm going to just put 6 bases here and give a particular sequence of the bases. So I'm going to put T, C, G, A, C, T. Those are my bases on the one strand. I'm going to need someone to help me with bases that we should have on the other strand. Anyone? can just unmute. Remember, how are we going to find these bases? As much as we are having two strands now, these two strands were once bonded together by weak hydrogen bond. So you need to tell me the bases that we find on the other strand because they were once paired. So what is the partner that will be paired with T?
Adenine.
A. Then the second base will be
Guanine.
G. The third one,
C.
Cytosine.
Cytosine.
C.
Cytosine.
Fourth one,
Thymine.
Thymine.
T.
Thymine.
Then
Guanine.
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 G. Then the next one.
Adenine.
Adenine.
Perfect. Thank you, guys. Now, let's remember point number 3. Point number 3 said both strands, I said you can use the word both or each. Both strands are going to be used as a template to form new strands, or each strand. If we're saying each, that means we're actually using both, are going to be used as a template to form new strands. So we are going to label these strands together. So these original strands that we've drawn now, we are going to label them. We will label this one as a template strand because we said we are going to use it as a template. Also this one, because we said we are using both, we are also going to label this one as a template strand. Now, in other textbooks, it will label this strand as coding. It will say it's a coding strand. It still means the same thing. We're going to use a code. We're going to use the sequence of the bases. When we speak about a code, it's just the sequence of the bases. We can also label this one as a coding strand. Now, when we look at the second point, I think 3 was the one with weak hydrogen bonds. Then obviously we speak about both strands being used as template strands. The next point, which was point number 4, it says that we use free floating DNA nucleotides. The key word there is DNA nucleotides. And where are we getting these DNA nucleotides? We are getting them in the nucleoplasm. The jelly-like or fluid-like substance filling up the nucleus. Look at these two words on the diagram, nucleoplasm and cytoplasm. Cytoplasm is the plasm of the cell. Nucleoplasm is the plasm of the nucleus. So this is what we are focusing on. Please don't make a mistake and say cytoplasm. Now it says we use free DNA nucleotides, meaning we are having these DNA nucleotides floating in the nucleoplasm. Now, it is important for us to specify that these are DNA nucleotides because in the very same nucleoplasm, there's also RNA nucleotides, which is what we are not focusing on here. So how are we going to identify which nucleotides is RNA and DNA? The sugar will be different. So the DNA nucleotides will have a deoxyribose sugar and the RNA nucleotides will have the RNA sugar. And then obviously uracil will be part of RNA. Now we use these free nucleotides to form complementary strands. How are we going to form these complementary strands? The free bases, DNA bases in the nucleoplasm are going to form a strand. So a free base that pairs with T will be bonded with T. So enzymes are controlling this. I think 5 years ago, you needed to know the name of the enzyme, but now you don't need to know the name of the enzyme. So as the bases move around in the nucleoplasm, a base that is paired with the exposed bases of both strands are going to pair with that particular base. So I'm going to change my color. What is the goal here? The goal is to form complementary strand. What does the word complementary means? The word complementary is telling us that the bases that are going to form this particular strand will need to complement the exposed bases. So it won't just be random bases. Any base that feels like bonding itself or attaching itself will go there. It won't be that. So the bases that we are going to have here are going to complement the bases that are exposed. So meaning if we are having a T as our first base here, a free DNA nucleotide that contains the adenine base is the one that is going to go boom and get bonded with that T. So that means it will be T and an A. Then a free nucleotide with a G, a free DNA nucleotide with a G is the one that's going to go there. And then here will be a C, and then here will be a T, and then here will be a G, and then here will be an A. If I make a mistake, please do rectify. I've been teaching since 6:30 in the morning. So if I mess it up, just please unmute and tell me where I messed it up. So, but I feel like this strand is fine. Now, how is it becoming a strand? It's controlled by enzymes, and these nucleotides are going to bond with each other and form a strand. And remember, we're saying it's a nucleotide, but we're showing you a base because you're not going to see the sugar and the phosphate. Now, the same thing that is happening to this strand over here on the left, this left template strand, is the same thing that is going to also happen in the template strand on the right. There are bases that are free there. Now, free DNA nucleotides will then pair themselves with the exposed bases. So we are using the template strand to say, oh, in the template strand, there's an A. So we need a base that is going to complement that A to be in that complementary strand. So then what happens here? We then get a T, a free thymine base in a DNA nucleotide is going to pair itself there, and then a C, and then it is a C here, and then a G, and then an A, and then a C, and then a T. Now, I used the color orange for my new strands here. The strands that are formed using the free DNA nucleotides, and these strands have bases that complement the exposed bases. These strands, we call them complementary strands. Complementary strands. That's what we had in point number 5, I think, when we were checking if point number 3 and 5 are the same or not. So now we are calling them complementary strands because the bases in these strands are complementing the bases that we used in our template strands. Now, remember, the goal is for us to form 2 identical molecules. We use DNA to make a copy. Now, in one of the points, it says the 2 molecules are formed, and each molecule contains an original strand and a complementary strand. Let's look at that. Now, they've said what? Each molecule, this is a DNA molecule. And this DNA molecule is made up of what? An original strand. Remember, this template strand was the original strand. And it is also made up of what? Of a complementary strand. That is one DNA. That is DNA 1. Then this is another DNA molecule now. This will be DNA molecule number 2. And this DNA molecule is also made up of 2 strands, being the original strand, which we labeled it as a template strand, and the complementary strand. Now, the goal is to have 2 identical DNA molecules. Now, how are we going to know whether the molecules are identical or not? We look at the sequence of bases. Let's check. Now, in terms of the bases that we have in DNA 1, which is what we've labeled as DNA 1, it's a T and A, C, G. It's the same thing here, T, A, C, G. And let's look at the bases. Let's continue. It's a GC this side, GC, AT, AT, CG, CG, TA, TA. We've achieved our goal. We have 2 DNA molecules. Each molecule now has an original strand and a complementary strand. I'm happy to take questions. If the points didn't make sense, the diagram normally does the magic. Let me come to people here. Please do raise your hand so that I can call you out and then listen to your question. Or you can just unmute and ask me a question.
Ma'am,
Yes.
I wanted to find out what's the title of this diagram.
DNA replication.
So when will we be required to draw this diagram?
You will not be required to draw the diagram.
Oh,
We are
okay.
drawing the diagram for learning purposes.
Okay.
So once we draw it like this, it makes way more sense. You can answer any question paper.
Okay. Thank you, ma'am.
All right. Let's go back to the slides, guys. There's something I want us to make sure that we remember. At the end, we have two DNA molecules. I need you to include the word molecule. You cannot say you have two DNA strands. That is incorrect because we are having 4 strands. If we are looking at the strands now, there's actually 4 of them. So you can't say you have 4 DNA strands. That will be 100% incorrect. So let's please try our best to look at the words and look at the terminology, because if your terminology is incorrect, yeah, unfortunately, we are going to penalize you. But I think I always feel confident after doing the diagram because you are able to see the whole process and you're able to see that these are 2 DNA molecules. That's how it's explained and that's how we teach it. So I'm trying to go back to the slides so that we can look at the points and you make a note of some of the things that I know learners normally make mistakes in. But if you have a question, I'm trying to change my screen now. So if you have a question, you can ask. I do have a minute. It's probably going to take me a minute to change the screen again. Okay, there you go. 7 seconds. It should be back. The notes. I'm back to the notes now. So if you can see the notes, thumbs up. It does tell me that the notes are shared. So thumbs up if you can see the notes. Okay, that means you guys can't see the notes. Please thumbs up if you can see the notes. Okay, cool, perfect. Thank you, guys. I was not seeing any reaction on my side. So let's go through this again. DNA, double helix unwinds. There's a tick. Hydrogen bonds between the 2 strands break and the molecule unzips. Each or both original strands serve as a template to form a new strand. Free-floating DNA nucleotides in the nucleoplasm. Please add this. Put a cross and write cytoplasm. For you to remember that you should not be confusing the nucleoplasm with cytoplasm. And also, please make a note here. Please make a note here, put an X and write RNA nucleotides. We are speaking about DNA nucleotides here and not RNA nucleotides. Then A will be paired with T, G will be paired with C. The original strands act as a template to form the new complementary strand. I think now, I don't think we made a huge mistake with this. Let's write this point again. I will check again, but let's write both 3 and 5. Let's not substitute. Even if, yes, it does sound like it's the same thing. It is the same thing, but let's just write them. I don't want to mislead you guys. I'm just going to try and figure it out to think about it. But here we are just speaking about a new strand. And here they are giving us the word complementary strand. Then each molecule, I need you to highlight this word. We are having a new molecule. Each molecule, if you don't want to use the word each, you can say both molecules because there are 2. They now consist of 1 new strand and 1 original strand. If you don't write the word original, you write template strand. that is also accepted. Remember point number 7, there is no mark. But now what happens? Each new molecule will twist back to form a double strand, from having 1 to 2 molecules twisting back. This results in 2 genetically identical DNA molecules. Please do write across and write strands so that you remember that these are DNA molecules and not DNA strands. The process is controlled by enzyme. There is no mark for that. All right. 6 and 9. This is after the molecule unwinds, and then the bonds break, the molecule unzips, free DNA nucleotides attach themselves to the exposed bases of the template strands. At the end, you end up having 2 DNA molecules, one made up of an original strand. The original strands are on the outside, original coding template strands, and the strands in the middle are the complementary strands. These are the strands that we formed using the complementary bases. This we've done. Let this be our question. You should have this question on your notes. So we've got 10 minutes left. Actually, the lesson should have ended at 7:30 because it's supposed to be 1 hour, 30 minutes, but I normally just push to 2 hours. Let's answer this and call it a night. I'll be nice. 5 minutes while I get water. So is it fine if I project it like this? Are you able to see the questions on your side? How do you guys want me to project it? Is it fine like this? Please just unmute and respond so that I know what to do.
Can we see the
No.
questions?
You are sorted.
Oh,
We saw the
Questions
top
are
part.
visible.
Okay. So what should I do? Is it fine if I send a picture to the group, picture of the questions?
Mm-hmm.
Is that fine?
Yes,
Yes,
it's
ma'am.
better like that, man.
Okay. Just give me a second. This is November 8th. Okay. I'm not sure if I zoom out a little bit, if it will be better. I think that's better. I think so.
Take it up a little bit. Yes,
And
thank you.
is it better? Okay.
No.
Hmm.
What happened?
It's
Zoom in
zoomed.
again.
Yes.
Yes, thank you, madam.
Oh, let me get water. Let me get water. 2 minutes left. Time flies.
Table 1. Oh, table 1 and 2. Just say to thermine guanine. What the fuck should I know? Cytosine.
Dude, your mic is unmute. We can hear you.
2 more minutes. Can we start marking, or do you need 1 more minute? 1 more minute? Or can we mark? Yes, no?
We can mark.
mark. Okay, thank you guys. Okay, let's do it quickly. Name the process represented in the diagram above. Can I see your hands? Can you go hands up, hands up, hands up, hands up. Oh, wait, two.
DNA replication.
Thank you. DNA replication. Name the part of the cell when this process occurs. I'm seeing the same people, guys. You see, there's also a Teams link. I can see the person, but I can't see the name. It says HTTPS Teams something. So
It's me.
it's you,
It's yeah, Carabello.
Carabello. Oh, okay, it's not written here. Carabello, yes, please do give us an answer.
Interface.
Name the part of the cell cycle when this process occurs. Interface, correct. 3, name the location in the cell where this process takes place. Sibabalo?
Is it not the nucleus?
Nucleus it is. You see this one, I am very interested in number 4. Number 4, I am super interested in. Let me even zoom it in a little bit. Give labels for 1 and 2. And I'm going to take different options. I'm going to take option A. Hopefully my pen is still working. And option B maybe for 1 and 2. And my option B for 1 and 2. I'm very interested in this one. Raise a hand, guys. Raise your hand. Raise your hand. Mohau?
I wrote. Can you hear me?
Yes.
Okay. For number 2, I wrote cytosine, and 1 I wrote guanine.
Okay. Thank you, Mohau. Anyone else with a different answer for the basis for 1 and 2? Anyone else? You can just unmute and tell me if your answer is different, then I'll take that as an option. Inga?
I have the same answer as me.
Same answers. Okay, thank you. Anyone else with different answers?
I have another different answer.
Okay, let me see who's talking. I can't see who's talking.
It's me, it's Mohau.
Mohau. Mohau, didn't you give us the first option?
Can I give you the second option? I just saw it right now.
Okay, give me the second option.
I would say number 1 is cytosine and number 2 is guanine.
Hey, we have to vote, guys. We gotta vote. Which one is it? Which one is correct? Tell me, unmute and tell me. Is it option A or option B that is correct?
Option A.
Option B. Option B.
Option A.
Option A.
All right. Thank you. Let's mute. 1 is definitely G. And 2 is definitely C. So Mohau, what made you change your mind?
Okay, this is a DNA replication, right?
Mm-hmm.
And the original strand on the left hand side, it's written CTG, and we know cytosine, its base pair is guanine. So coming to the right hand side, the original strand is the one that has 1, right? And we saw from the left hand side that the original strand had C. So on the right hand side, which means uh, the first, the first base would be C. So that's what made me change my mind. But then again, I can see that I forgot the star mine. The
Okay.
parent is star mine.
So this is a very common mistake in DNA replication. So that's why I had 2 options, because I knew I'll probably get 2 possibilities here. So your first part of the explanation, 100% spot on. Now, these outer strands here are our original strands, our template strand. So if we look at the base that is attached to the template strand, it's a C. So that means this C was initially paired to this base over here, which is also attached to the original strand. So the C was initially paired with the G. So that's how we get the 1. And then this G would be complemented by C, which is our 2. So there's different ways of figuring this out, but I think that's the most simplest one. So that means option A is the correct one, not option B. Then state 2. Okay, I no longer have the slides now. They vanished, so just give me a second. Okay, state 2 ways in which the structure of RNA differs from the structure shown in the diagram. Julian?
Okay, so way 1 is that RNA, 1 strand is used as a template, while DNA, 2 strands are used as a template. Then way 2 is that RNA contains ribose sugar, while DNA contains deoxyribose sugar.
Okay. I need you to give me your point number 1 again.
Okay, number 1, 1 strand is used as a template, or it's single-stranded, basically.
Those are 2 different things. So tell me which one you
Okay.
answered here. So saying it's single-stranded and 1 strand is used as a template, those are 2 different things.
Okay, what I wrote that 1 strand is used as a template.
And then you said in DNA, there were 2 strands.
Yes, ma'am.
So for the same point, you then said DNA, we are using 2 strands. Okay. Then your option number 2.
I said that RNA has ribose sugar.
A has ribose sugar. And then DNA
has deoxyribose sugar.
Is deoxyribose sugar. Okay, so out of 2 marks, you are getting 1 mark. And that 1 mark is for the second comparison. DNA has deoxyribose sugar. RNA has ribose sugar. That's a mark. For this option is wrong. Because I'm trying to figure out RNA 1 strand is used as a template. As a template to do what? It's used as a template to do what exactly? So I just want to understand your understanding. So just explain it to me so that I know what I'm rectifying.
I don't know if I'm confused, but that template being used for, I think, translation to when the mRNAs has to correlate with the tRNA.
Oh, okay. So you are bringing in protein synthesis.
Yeah, in that sense.
Okay. So the question here, I want us to read and understand it. State 2 ways in which, what exactly? This is our keyword, in which the structure of RNA differs from what? The structure of DNA. We are focusing on the structure.
Ma'am, can I
So-
give my answer?
I didn't hear.
I said, can I give my answer?
Let's listen.
Okay, I wrote, the question is state two ways in which the structure of RNA differs from the DNA structure shown in the diagram. I said that the RNA structure is single-stranded and the DNA structure is double-stranded.
That would be correct. So when I was getting this first option, I'm gonna go back to Teams. I don't remember who was talking. Was it Lebohang who was giving me the first answer? It was, I can't see now who was talking. Oh, it was Julian. Yes, it was Julian. So she said something about one strand is used, then you said one-stranded molecule or a single-stranded molecule. So if you say it is a single-stranded molecule and DNA is a double-stranded molecule, that is correct because you are describing the structure. Even if you speak about protein synthesis and translation, there is no way where we use RNA as a template. We don't use it as a template. Now, I think I see where the confusion is. If we move from DNA to form a messenger RNA strand, we use that messenger RNA strand to form a transfer RNA strand. I see where this is coming from, but we'll address it next week, right?
Okay.
Yes?
Yes, ma'am. I got you.
So I definitely see where this is coming from, that when we form a transfer RNA, we use one strand, which is from the messenger RNA. But that would not be the correct answer for the question that we are given. We are just comparing between an RNA structure with the DNA structure, the basics. This one is single-stranded, that's the RNA. DNA is double-stranded. This one has ribose sugar, this one has deoxyribose sugar. In terms of bases, RNA will have a thymine base and DNA will have RNA will have a uracil base, DNA will have a thymine base. You can also compare the length of the structure. So RNA is normally shorter and DNA is normally longer. So those are the points that we can use to compare the structure. Now, DNA replication and everything that we are mentioning here is easily confused with protein synthesis. That is common. So even if you are confusing it, don't think you are alone. It is a common thing. It's past 8 o'clock. Yes, you can ask a question.
Can I ask that how do we see that in DNA it is a deoxyribose sugar, because the question says as shown in the diagram?
Let me go back. As shown in the diagram. So yes, that is a good argument to say we're not seeing the sugar. So then we can say, okay, cool, we're not answering it. The sugar answer cannot be part of our options. If they've said shown in the diagram, that is a good one. So if it just ended here, we could include sugar, but this becomes an argument every single year at the marking center to say, let's not penalize learners. Even if a learner is saying a difference about the sugar, they are showing understanding to say, oh, okay, cool. DNA will have this type of sugar and RNA will have another type of sugar. 100% agree. Can we see it from the diagram? No. So we shouldn't include that to be safe, because we don't know what arguments are going to be there this year. There's no time to do this one. Absolutely no time. So if you do get time, I will send a memo on the group. And then there's other activities that I'm going to send. Please do the DNA replication activities from that document that I'm going to send, because you can see the next part that we're going to do is protein synthesis. And I need to probably come back and sleep and then get ready for the lesson, because this is a lot. Protein synthesis is a lot. So I'll send a memo on the group for this activity. And if you have questions, please do ask questions. Like I always say, my responses will be delayed due to my work. I do work 8 hours. If sometimes it's 10 hours at work. So my responses will be delayed, but I will answer. So if there's anything unclear or anything that I need to check on my side, if there's something I don't know, I'll just double check with my colleagues and then come back to you guys. I see someone's hand is up. Sibabalo? Okay. All right. Any questions or comments or anything that you need clarity on based on what we've covered today? Yes, I'm listening.
Yes, ma'am. So when you give us like work to do we send the answers in the group or do we send them directly to you?
So normally you can send them to me if you feel like you need a memo before I share it on the group, but it's not possible for me, especially during the week, to go through the activities and then mark everyone. So if you need a memo, you can send the activity to me or just request it, then I'll send it to you. So it's up to you. What I normally prefer, if people want to discuss something on the group, so then you can say, hey guys, these are my answers. Then you guys can have a discussion. I'm fine about it because now there's probably plus minus 20 of you. I know in the group there's also parents, so there's plus minus 20 of you. So we don't want a situation where everyone is just sharing their answers and other people are going to feel shy to share their answers. But discussions, I'm happy for you guys to have discussions on the group.
All right, thank you.
All right. Anyone else? Oh,
Yes,
anyone else? Yes, is it basically yes?
can you please like give us like last year's practical for the strawberry thing for the write-up work?
What would form part of the write-up work? I'm trying to remember.
We haven't done it yet, but it's an upcoming test. So something to use to practice.
I'm gonna have to try to remember what was done last year. At my school, we do things differently. So we don't follow everything that the government gives us. So if you did something about strawberry at your school, we could have probably used a banana or something else. So I'll check online. So we don't follow anything. Most of the things actually that the government gives us. We don't even write national SBAs. So we do our own things. So I'm gonna try and figure out. You can remind me on the group, probably on Friday. Then I will see what I can get. Are you doing the DNA extraction at school?
Yes.
Okay, so for that, you can even watch a video on YouTube, strawberry DNA extraction, just to understand how you get to extract it. In terms of the questions, there's normally a drawing there. So you need to follow drawing rules. That's what I'm trying to remember. They normally ask you things like, why did they use methylated spirit or alcohol? Why did they use a dishwashing liquid? Why do you need to mash the banana or the strawberry first? They're basically questioning you about the different steps of the experiment. That's what I'm trying to remember now. So if I find anything, I will send you whatever that I get. But if I don't find anything, then I won't have anything to send.
Okay, it's fine.
But those questions are what's coming to my mind. Those are what some of the questions that I remember.
Okay, thank you.
All right, thank you. Okay, guys, let's call it a night. Remember, before you leave, you unmute and you say "Totsins, bye," and then have a good evening, guys. Thank you so much for taking time to join the lesson. Whew, it's been a long day. It's been long. From 5:30 in the morning. Okay.
Yeah, very long.
Thank you so much, guys. And good night.
Good night.
Bye.
Bye.
Bye, ma'am.
Bye.
Guys.
Bye.
Bye. Bye. Bye. Bye, guys.